Yes. It says I cannot share my. Okay. Started like this. All right. Hi, good morning, everybody. My name is Xiaoriya. I'm a senior student from biology department. Today, I'm going to talk about my research project. Can recovery from repeated heat stress event vary by sex and habitat? For plants to grow and reproduce successfully, they have to grow within an optimal temperature. Any temperature that fell outside the optimal range will cause stress, which could cause damage to the plant. However, with accumulation of stress, plant can actually recover ability to recover from any damage caused by stress, which referring as priming. Just like the graph I'm showing right here, when plant get forced heat stress, they could got really bad damage and it will take them really long time to get recovered. However, with second heat stress, they could actually recover faster and not get as much damage as before. So I hypothesis for my study that plant with more exposure to the heat will actually recover better than the less exposure plant. The plant we're using today is called Marchantine inflexa. We're collecting from Trinidad in two different habitats. The first habitat is more closed field. We call that shaded plant, referring as ETR right here. The second plant we collected from a different habitat, which is more open field. We call that sun plant, referring as NOR. So in my study, the method we're using is we have collecting 20 different genotypes, 5 female, 5 male from each two different habitats. Then we assign 20 different genotypes into two groups, experimental group and control group. The experimental group will get 2 different heat stress events. Control group will only have 1 heat stress event. For the first heat stress event, we will use 50 minutes for 50 degrees Celsius. For second heat stress event, we would do 55 minutes for 50 degrees Celsius. And then we would collecting the efficiency of photosynthesis for each different genotypes. But before we collecting, we would put plant right here in a clip. We call that dark treatment. The purpose of putting plant in a dark treatment is for the plant to reset the fluorescence they reproduced. That way we can get the most accurate data. So after we did the master part, we finally collecting the data. Our data indicates that there is a strong significant difference with the number of exposure to the plant. So the plant with more exposure do actually develop prime better and recover better than the less exposure one. Additionally, we have a sexual difference right here indicates that female actually recover better than the male plant. And right here, we also have a habitats different. However, it's not as significant as two other trials. However, my data still supported that sounded plant actually recover better than the shaded plant. So in conclusion, my hypothesis was actually strongly supported. Plant with more exposure to heat stress would recover faster and better than the less exposure one. Additionally, there is a sexual difference could consequence survival ability for plants to survive in a warm climate. Thank you so much. That's all about my presentation. Please let me know if there's any other question. Thank you.
Thank you. That was very nice. Very good job.
Thank you.
Do we have any questions for Grace? Here we go. We do, right in the question and answer box. So Dr. Cooper is asking, do you think the same would occur with cold stress?
even though I haven't done research about it, but I think it might be because in my research, the plan we used was actually being tested with water stress. So since they're showing a similar pattern of recovery, I assume there might have a similar recovery better to the cold stress as well. Thank you for your question. Hope I answered that.
Okay, we have another question for you. Dr. Cooper says thank you first, and then Jennifer asked, what do you think may be responsible for the sex differences in the response?
It could causes by temperature. For my first trial, we actually couldn't get any sexual differences because the temperature we were using is actually 5 degrees lower than what we have right now. However, after my second trial, we increase the temperature to 50 degrees Celsius. We can see significantly sexual differences in my result part. So I think it's mainly just because of the temperature control. Thank you.
Any other questions? Okay, thank you very much. Good job.
Thank you.
Your next presenter is Sahana. Are you ready?
Yeah, I'll share my screen.
Let me share your screen when ready.
Hi guys, my name is Sahana Holla. I'm a senior here, and I'll be presenting on the effect of carbonation on dehydration-induced fluid intake in female rats. Now, carbonated beverages are a very popular beverage choice, especially in Western cultures, but it's not well understood on how it interacts with our full body fluid balance. So recent studies have begun to look into the effects of carbonation on effects of carbonated beverage consumption on our body, and they found a few things. They have found that increased carbonated beverage consumption leads to an increase in release of ghrelin, which is a hunger hormone, and can then promote weight gain. In addition to that, CO2, carbon dioxide present in carbonated beverages, are known to stimulate oral receptors that are also sensitive to cold water and associated with increased satiation. So when we take these findings together, it then suggests that carbonated beverages could be associated with increased satiation. So our goal for this series of experiments was to test the hypothesis that carbonated fluids would be more satiating than non-carbonated fluids during certain thirst challenges. So for the study, we used 15 female long evidence rats with access to food and water, and their body weights were monitored daily. The experiment itself, or the layout, could be separated into 3 different phases. So week 1 was the habituation week. So here the rats were given the full week to acclimate to the new environment. Week 2 was specifically acclimation to methods. So we gave them access to the water bowls, which were separate water bowls that we placed at the front of their cages that we'd be using during testing. We also gave them access to the carbonated water so that they'd get used to that sensation. Week 3 onwards is when our testing began. And testing involved 3 different experiments, all modeling different forms of dehydration. So our experiment 1 was a 24-hour water deprivation, which is a combination of an osmotic and hypovolemic dehydration. For this, water was removed from the cages 24 hours before the test time. And then on test day, either a bowl of carbonated or non-carbonated water was given back to the rats. Experiment 2 was a hypertonic saline treatment. And this was a osmotic dehydration. For this, water and feed were removed the day of the test. Then the rats were given an injection of lidocaine and then also hypertonic saline. And then 15 minutes after the injection time, the carbonated bowl or the non-carbonated bowl of water would be given back to the animals. The last experiment, the isoproterenol treatment or hypovolemic dehydration, was done similarly to the hypertonic saline treatment, where they were given an injection of isoproterenol, and then 15 minutes later, the rats were given either a bowl of carbonated or non-carbonated water. Now, all these experiments were done in a counterbalanced manner, meaning that in the first day, half of the rats would receive carbonated water, the other half would receive non-carbonated, and then 3 to 4 days later, they would switch the fluid type. In addition to that, all carbonated or non-carbonated water was made by spinning the carbonated water as well, and then the fluid intake was measured for 20 minutes. Then the intake was analyzed using t-tests, and also we ran a 2-factor repeated measures ANOVA to look at the body weight changes during experiment 1. As expected, all of the experimental manipulations did result in a drinking response, but there was no significant difference between carbonated and non-carbonated fluid consumption during either the 24-hour water deprivation, hypertonic saline treatment, or the isoproterenol treatment. And these are shown in figures 3, 5, and 6. Figure 4, however, is showing us just the difference between the average body weights the day before the 24-hour water deprivation and the day of, or the day that we're doing the test. And it does show that water deprivation did decrease the body weights, all in all kind of showing that our manipulations did work and they would induce a drinking response. Since there was no significant difference between the carbonated and non-carbonated fluid consumption, this does suggest that carbonated fluids would be equally satiating as non-carbonated beverages. But that doesn't necessarily mean that there aren't any subtle differences that we can't necessarily see in this study. Since we did use water bowls instead of bottles with a spout, we were unable to use ligometer testing. And ligometer testing would have allowed us to look at further microingestional differences. So for our future studies, we would like to conduct ligometer testing to look at variation in burst number and burst size. By doing this, we'd be able to better distinguish between satiety and hedonic preferences. Hedonic preferences meaning whether the rats just enjoy the flavor of the carbonated water or the feeling of the carbonated water instead. In addition to this study was only conducted on female rats. And we know that there are sex differences in fluid intake due to estradiol. So our next step would also be to examine the effect of carbonated water on dehydration-induced intake in male rats, and perhaps see if there could be a difference in carbonated water intake between male and female rats. Thank you. So if you guys have any other questions about that, I'd be more than willing to answer them.
Thank you. That was very good. So we have a couple minutes left. Does anyone have any questions? Put them in the Q&A box, please.
In juveniles, I'm not sure. When it comes to sex differences, I'm not 100% sure on when the estrous cycle and stuff starts in juveniles and rats. So I can't say whether sex differences would be there, but I want to say, I feel like I would need more information on when all the cycling begins to be able to answer that question with sex differences. But I'm not sure if there would be a difference in simply drinking, like difference between carbonated and non-carbonated consumption in juvenile rats. I hope that answers your question.
So are there any other questions? Thank you, Dr. McClatchy. Oh, wait. I see. Okay, so Jennifer asks, do you think the temperature of the water may affect fluid intake of carbonated versus non-carbonated?
Temperature of the water. I didn't think about that. Maybe actually. So since we do know that carbonated water and cold water do lead to stimulation of the same oral receptors. So I do wonder that maybe if the carbonated water was colder, we could test it that way too and see if we were to cool both the non-carbonated fluid and the carbonated fluid, if we would see a bigger response because of further stimulation with the carbonation as well as the cold temperature.
right. Give them one more. Okay, she says thank you. Dr. McLechty did too as well. So I don't think there's any more questions. So thank you very much. Great job.
Thank you.
So our next presenter is it Ayanna?
Yes, ma'am.
Okay, and you may share your screen at any time.
Hi, my name is Ayanna Adaniel. I'm a senior, and I'm presenting on my topic, which is how sex, habitat, and relative humidity influence germination of asexual offspring in a tropical plant. So in this case, the tropical plant is a liverwort called Marchantia inflexa. So most studies that focus on the relationship between water requirements and plants focus mainly on liquid water. So very few studies have focused on water vapor or relative humidity. And relative humidity can be very important to a plant, and species can respond differently to varying humidities. This is especially important if relative humidity changes germination responses, because germination is important for colonization and establishing populations. So to determine if relative humidity influences germination, we varied relative humidity in the air above liquid water to test if, we varied relative humidity in the air above liquid water to test if germination differed by sex, and 2 habitats from Trinidad and Tobago that varied by moisture. We used a plant, Marchantia inflexa, because a past study had shown that this plant demonstrates water stress responses by sex and habitat. So we thought that if we varied relative humidity similar, the results could be similar. So we had 2 relative humidity conditions. We had a high at 100% and a low at 65%. And we predicted that the high relative humidity condition would produce more germination. We had 2 habitats, like I mentioned, a dry, more exposed habitat indicated on my poster as NOR, and it stands for North Oropouche River, and a wet, more exposed or less exposed habitat indicated by ETR, and that stands for East Terare River. And so we predicted that plants from the drier habitat would produce more germination and be more sensitive to changes in relative humidity. And lastly, we had 2 sexes, male and female, and we predicted that females would have higher germination or produce more germination than males. Like I mentioned, we had 2 habitats. We had a wet habitat, which is ETR, and a dry habitat, which is NOR. And these are just pictures of the aerial view of these habitats. We took 18 genotypes from the habitats and grew them in common conditions. So the asexual offspring of Marchantia inflexa are called gemmi, and they're pictured right here. And for the high relative humidity condition, we took each genotype and placed it in a test tube filled with water. So the gemmi just sat right on top and were floating. To control for relative humidity, we used a container filled with pure water. So in the high relative humidity condition, it was the container filled with water and 18 test tubes filled with all the genotypes. And all of this was sealed in an airtight container. For the low relative humidity condition, it was essentially the same, except we used a salt solution to control for relative humidity instead of water. We used water and magnesium nitrate. And so between the 2 conditions, temperature was the same and only relative humidity varied. So the gemmi were allowed to grow for 2 days, and after 2 days, they were stained with methylene blue. Methylene blue is just a dye that killed the gemmi and stained the rhizoids. So the rhizoids are analogous to roots for gemmi and are also indicative of germination. So 3 trials were performed, and we determined that the high relative humidity condition produced more germination than the low, which suggests that Marchantia inflexa prefer high relative humidity. We found that females germinated more than males, which suggests that females might have a quicker growth rate than males, or from an evolutionary standpoint, females might need to germinate faster in preparation to mature offspring. And lastly, we found that plants from the drier exposed habitat, NOR, germinated more than those from ETR. And this also suggests that plants from NOR might germinate faster when conditions are more favorable for them. So plants from NOR were used to having little moisture in their environment. And so when they were subjected to the high humidity condition and there was more water available, they grew because conditions were more favorable for them. And so overall, as I mentioned, more germination was associated with high relative humidity, females, and plants from NOR. The results support the idea that relative humidity influences germination, and also supports the idea that the gemmy are sensitive to differing air conditions despite floating on liquid water. So that's it for my poster. If anyone has any questions.
Thank you. That was very nice. Do we have any questions out there for Ayanna? I liked how you moved around the screen so we could see that. That was really neat.
Hopefully I didn't move it too much.
Oh no, it made it very readable. It's a good job.
Thank you.
Hey, Ayanna, this is Jessie. I'm just gonna ask you a question. What was most surprising from your project?
Just how similar the germination rates were. Like it was the high relative humidity condition did germinate more, and females and plants from NOR did germinate more, but I definitely thought that there would be more of a difference. As you can see, they're pretty similar, but still the difference is not as drastic as I thought it would be. I hope I answered your question. Thank you.
We actually have two questions now in the Q&A. The first one is from Kay. Do you think there might be a sex difference in germination between ETR and NOR?
Yes, I definitely think so. So we separated it and the analyses. So there's definitely a sex difference. And I think it's due to probably the female reproductive processes needed for reproduction, and just the differences between females and males overall. What was the other question?
Dr. Cooper asked, does the, I don't know this, MG2+ maybe block ion channels?
Oh, that is such a good question. Personally, I haven't thought about that, but that would be very interesting to look into. Yes.
Next project.
Thank you.
Okay, let's see. And then Dr. McLetchie asks, do you think that more, is it Jemay, will germinate given more time?
Yes.
Sure I said that right.
Yes, I definitely think the Gemmy will germinate if we gave them more time. We actually conducted a pilot study at the beginning to determine how many days to let the Gemmy germinate. So we decided on 2 days because that was enough to provide like a difference between germination. So if we left it longer, they all would have germinated up to like 100%.
And then did you see Dr. Cooper's response?
Oh.
It can block. Well, thank you very much. You did a great job, Ayanna.
Thank you.
Thank you, everyone.
Hi, let me screen my share my screen real quick.
Okay, thank you. You go ahead when you're ready.
Okay. Okay, hi, my name is Katelyn Fields and I am a senior biology major here. And the focus of my research is the effect of melatonin on the morphology and biofilm formation of Klebsiella aerogenes. And this is a gram-negative rod-shaped bacterium found in the human gut microbiome. And what is also found in the human gut is melatonin. And while we usually think of that as an important neurohormone in charge of the sleep-wake cycle, it is also found in the human gut. And previous work by my lab has shown that Klebsiella aerogenes actually shows sensitivity to melatonin. So the overall purpose of my experiments were just to determine exactly how Klebsiella aerogenes is affected by melatonin and just the different characteristics of this interaction. So I studied this overall purpose with two different experiments. First, I looked at the morphology of Klebsiella aerogenes during its exponential and stationary growth phases in the presence and absence of melatonin. And then I also studied the biofilm formation of Klebsiella aerogenes in the presence and absence of melatonin. So just for a little bit of background information, morphology is just like the individual shape of each bacteria, and every type of bacteria goes through this basic growth curve here. And the actual phases that I studied were this exponential or log phase here and the stationary phase. And then during the log phase, that's when the bacteria are rapidly dividing. And then during the stationary phase, the growth rate is equal to the death rate, and that's due to the nutrients of the culture being used up. And then for the biofilm experiment, biofilms are basically when bacteria excrete extracellular polymeric substances to help with adhesion, and they just aid in the pathogenicity of the bacteria because they just allow the bacteria to adhere to different surfaces. And for the biofilm experiment, I actually studied wild type Klebsiella aerogenes, but I also studied two different mutants, which have a mutation in a gene called FIMD. And this gene has been shown to be responsible for the localization of adhesive fimbria. So just for a little bit of my methodology for the morphology experiment, I grew the bacteria in an overnight culture, both in the presence and absence of melatonin. And to determine the growth phase that they were in, I looked at the optical density. And then once the desired phase was reached, I plated them, grand stained them, and then looked at them in a microscope to produce these images here. And then I used a computer program to measure the lengths of the bacterial colony, just the individual bacteria. And then for the biofilm experiment, I used a 96 well plate and inoculated it with the 3 different strains of bacteria, wild type, and the 2 mutants. And I grew those both in the presence and absence of melatonin, and also grew some blanks as well, just for reference. And I used a plate reader to measure the optical density of the biofilms that were formed, and I stained the biofilms with crystal violet. So here's an overview of my results here. I wish I can't zoom in, but either way, I'm sorry. But for the images of the different bacteria, these are just sample images, and this column here is for the bacteria in exponential growth. This is during stationary growth, and then this row is in the absence of melatonin and in the presence of melatonin. And just from like briefly overlooking this, you can see that it's quite obvious that the bacteria in the exponential growth phase appear to be longer than in the stationary growth phase. But it's kind of hard to tell the difference between melatonin and no melatonin. So the results are quantified here in this box plot. And as you can see, there was a statistically significant difference both in the absence and presence of melatonin in terms of growth length between exponential and stationary growth. So in both cases, the bacteria were statistically significantly longer during exponential growth. And then interestingly, there was a significant difference between stationary growth when there is an absence and presence of melatonin. So the bacteria during stationary growth in the absence of melatonin were significantly longer than the bacteria in the presence of melatonin. And then for the biofilm experiment, the data produced by the plate reader is summarized here. And in both melatonin groups, presence and absence, there was a statistically significant difference between the 2 mutants and the wild type strains. So that just shows that, and that just means that the wild type bacteria produced significantly more biofilm than the 2 mutants. So that suggests that the FIMD gene is required for proper biofilm formation. And that's just because FIMD helps make those adhesive fimbria. And then while there appears to be somewhat of a difference between the 2 wild type strains in the absence and presence of melatonin, it wasn't exactly statistically significant because the p-value was 0.0541, but it was almost significant. So the group in the presence of melatonin produced more biofilm. And I think if I were to be able to repeat the experiment, perhaps that significant difference would be found. But overall, my research just allowed me to learn more about Klebsiella aerogenes and how it reacts with melatonin. And thank you so much for listening. And please let me know if you have any questions.
Thank you, Katelyn. That was a great job.
Thank you.
So if you have any questions for Katelyn, please post them in the Q&A box. So Katelyn, while we're waiting for a question or two, let me just ask you, what enticed you to get involved in research at the University of Kentucky?
Yeah, so I want to go to medical school, and I've always wanted to since the start of college. And while working clinically is different, I also wanted to get the more research side of science. And I'm really glad I did. It's really helped me to learn how to deal with challenges because a lot of things often go wrong. So it kind of helped me learn how to troubleshoot, and I really enjoyed my research experience.
Okay, and you're a senior getting ready to graduate?
Yes, ma'am.
Well, congratulations for that. Great
Thank
feat.
you.
So Kay, we do have a question for you. And so Kay asked, why might melatonin have an effect on the biofilm production? And then the second part of the question is, what would be the biological reason?
Yeah, so research done by my mentor, she actually sequenced the genome of Klebsiella aerogenes, and she found that in the presence of melatonin, stress response genes were actually downregulated in the presence of melatonin. So perhaps this downregulation of stress response genes allowed for more biofilm to be produced. So that's kind of my hypothesis, but I'm of course not sure. Good question.
Okay, Dr. Cooper asks, do you think if you incubated tryptophan, a precursor to melatonin, might it be the same effect?
Possibly. Probably, but I'm not sure, but that's a good question. I would think so, since it's like an amino acid and it would help with the growth and stuff, but again, I'm not sure.
Any other questions for Katelyn? Okay, good job, Katelyn. Thank you very much.
Thank you.
Hi. Hi, Natalie. You're our next presenter. When you're ready to go, you may share your screen.
All right, my name is Natalie Hahs, and I'm a senior here at the University of Kentucky, and my research was over the foraging behaviors in house sparrows in the face of uncertainty. And so for many animals, finding food has been a struggle. So acquiring energy has been a struggle for many animals, as they do face many challenges that they have to overcome, such as where to find food, what kind to search for, how to deal with any potential prey defenses, and then also the possibility of becoming prey themselves. And so animals must also make these foraging decisions with uncertainty in the quality, quantity, and location of food. And so this idea of uncertainty has received little attention, but there's been some study on the preference and how one may choose where to look. And so when faced with uncertainty, these foraging decisions can impact both parent and any potential offspring. An individual's preference may be variance sensitive by having a preference to forage in areas where food may be highly variable, which would make them more variance prone, or where availability is more uniform, therefore making them more variance averse. And so in this study, we observed parent house sparrows to investigate the variance sensitivity in foraging when providing food for young nestlings, and to identify if parent sparrows change their foraging decisions when conditions change. And so we hypothesized that the parent sparrow foraging preference will shift based on brood condition and food availability. And some questions that we had going into this was, can parent sparrows associate color cues such as the colors associated with the wells that you see in figure 4, or are there conditions that cause parents to shift between being variance prone or more variance averse? And so what we did was we took one adult male and one adult female per aviary and induced breeding, and that breeding was successful in 3 seasons from 2015 to 2017. And then these parent sparrows are then trained to flip the wells and lids and probe the sand for crickets to then be fed to their offspring. And so videos were recorded twice a day from when nestlings were 2 days old to when nestlings were 10 days old. And so each board varied by color-coded wells, and different levels of variance were uniform variance where all the wells had the same amount of food, medium variance where 2 wells had 6 pieces of food, 2 wells had 2 pieces of food, and 2 wells had 0, while the high variance just had 2 wells that had 8 pieces of food and 6 wells of 0. And so the location of food was randomized by color for each treatment for each pair of birds, and every 3 hours the boards were refilled, and the location and the number of wells per each color were re-randomized, but the colors associated with each variance type stayed the same. So if the initial color were associated with high variance was blue, it would have remained the same the entire duration of the recordings. And so the other color options were either brown or yellow, but whatever color was assigned to the variance type would keep that assignment for the duration of the recordings, as I said. And so at day 6 or 7, so age 6 or 7 of the nestlings, the food available was reduced for 40 hours, and it stayed that way for each pair of birds. And then once that time had elapsed, the food was restored to normal. And so in the video recordings, data was collected on sex, the time and location on and off the board, and at the wells, the behavior of flipping and digging in the wells, time food was found or the well was abandoned. And so we also noted if food was found or not before leaving a well. And so what we found from this data was that parent sparrows appear to be able to associate color with distribution, which signifies some sort of learning, and that is shown in figure 2. And so also searching behaviors shift as they gain experience, which also signifies some learning in the treatment groups. And then the probability of finding food decreases when there is a high quantity of food available, as shown in figure 3, but this was present only in the variable group, so the medium and high variance. And so this suggests that there is an impact on what parents expect to find while foraging. And we also used a multi-logistic generalized model that tests the probability when there is more than 2 possible outcomes. And what we found was that there was a significant decrease in the more variance parents being more variance averse with the increase in age. So they do become more risk-taking when the nestlings are older or whenever they have had more experience with the trials. But there is no evidence, no significant evidence that the two sexes had differences between the preferences on where they would forage or the ability of learning, the distribution. There's also no evidence that the nestling condition changed the preference of these parent sparrows. So far, and so because there was no difference in the sex between the sexes being more variance prone or variance averse, parent sparrows were more likely to choose the uniform distribution or the less variant option depending on the brood age and experience. And as they have gained more exposure and they were able to possibly learn, there is some evidence that they were able to use those colored cues to distinguish between the different variance types. And so far, I would say that these results don't fully support our hypothesis that behaviors would change based on the food availability and the nestling age. And so to move forward, we need to do more analysis to better understand these results and to determine the likelihood if a parent sparrow might return to a well immediately after just finding food, because this could possibly change the results that we have now.
Thank you, Natalie. A good job. So do we have any questions for Natalie? Oh, we do. Hang on just a second. Can you see the question as well? So it's from Dr. Cooper. So it says, do we know?
Yes.
Okay. Do you want to read it and then answer it?
Sure. Do
Okay,
we know the rhodopsins these birds have in their eye receptors, which would help know what colors they might respond to? It would be interesting to know if they can detect ultraviolet spectrum as some other birds. And that is a great question, because I don't think I talked to Dr. Westmead about this, but I did talk with some of my friends on how they're able to distinguish between the colors. And I looked into it a little bit, but I believe that birds can detect, is it 4 ranges of light? So humans can only do 3. And so birds can do that 4. And I believe I found that in some studies that I looked up based on my own interests.
Dr. Cooper says, thanks. Okay, so just a question in general, Natalie, how did you get started in research?
When I was a freshman, I did the STEMCAS program, and so that enabled us to get the undergraduate research experience very early on, and that was mainly one of the reasons that I wanted to come to the University of Kentucky, was to get into the research. But what kind of research, I only realized that recently, of which one I liked, which type I liked.
I tell a lot of my students all these great choices are a problem to have. Okay, I think that is all that we have for you today. You did a great job. Thank you very much.
Thank you.
Okay, I'm ready to start. Oh, it says I cannot share screen.
Hang on just a second, she'll give you access.
Okay. Okay, do you all see my screen?
Yes, we can. Starting.
So, hello everyone. My name is Bahar Katanbov. I'm a senior at the University of Kentucky, and I'm double majoring in neuroscience and biology. My research paper, actually my research was a review paper, and it was about the impact of traffic-related air pollution on the physiology of vertebrates, specifically their brain and nervous system. The 2 pollutants that I chose were lead and carbon monoxide. The first pollutant that I focused on was carbon monoxide, which is made when carbon-based fuels, for instance, wood, coal, and oil, burning completely. The other main pollutant is lead, and the two biggest sources of lead are ore and metal processing. I'm going to zoom in so everyone can read my screen. So lead does affect neurons and neuronal function by blocking N-methyl deaspartate receptors, which are the receptors that glutamate binds to. And as we all know, glutamate is an exciter neurotransmitter. It causes some structural damages, such as irregular nuclei, denaturation of myelin sheath, and reduces the number of neurons, transmission activities, and neuronal growth, and leads to extra sensitivity, slowed movement, and limb weaknesses. It does substitute for calcium ions and passes through the blood-brain barrier and interferes with synapse formation. Lead also affects brain and brain function by reducing the volume of brain, especially the prefrontal cortex region of the brain, which is responsible for decision-making. And it leads to some psychiatric conditions, some behavioral problems, and neuropsychiatric and hyperactivity disorders. It also damages cells within the hippocampus area, which is a formation in the brain that is involved in learning and memory. And it does accumulate fluid in blood-brain barrier and causes headaches, dizziness, nausea, seizures, numbness, loss of coordination, memory, and the ability to see or speak. On the other hand, carbon monoxide substitutes oxygen in bloodstream and leads to the formation of carboxyhemoglobin, partial systemic hypoxia, and toxicity. And it limits oxidative phosphorylation, reduces the amount of ATP that is produced in ATP synthesis, and reduces the oxygen carrying capacity since it substitutes for oxygen, and leads to generation of superoxide and damaging cells and tissues, which eventually results in dizziness, weakness, nausea, disorientation, and confusion. And as you can see, some of the symptoms are actually similar. However, in some severe cases, it has caused vomiting, collapsing, or even coma in rats. Unfortunately, to this day, the pathway by which carbon monoxide affects vertebrates' brain is still unclear. And surprisingly, there was some benefits associated with low levels of carbon monoxide, and that's it limits the apoptotic cell death in neocortex and hippocampus and reduces the damage of cerebral hypoxic and a stroke. It also suppresses myelin reactive immune cell activation inside the central nervous system and helps in the reduction of autoimmune neuroinflammation impairment. Some of the things I recommend for future studies, they can focus on the long-term impacts of constant exposure to traffic-related air pollution in crowded cities and how one's body would react to the presence of two or more contaminants at the same time. So as I said earlier, my research paper was based on findings from peer-reviewed primary research articles and meta-analysis by searching databases such as Google Scholar, Web of Science, PubMed, and UK libraries. Some of the keywords that I focused on were neuronal impacts of lead, impacts of carbon monoxide and phosphorylated oxidation, and sources of traffic-related air pollution. And all my sources were written in 2000 to 2020, with the exception of two sources, which are written in 1960 and 1988. And I mainly wanted to keep my paper written based on newer findings and newer sources. And I would like to finally thank my research mentor, Dr. Keisha Noy, for giving me the opportunity to conduct research in the middle of pandemic and write this paper.
Good job. Thank you. Important acknowledgements there for sure. Do we have any questions?
Sorry for the voice crack. I woke up sick, so.
Very well. So we're waiting on just maybe a question or two that will pop in because sometimes it's a little delay. So why don't you tell me how you got involved in research at the University of Kentucky?
So I always wanted to conduct research at UK, especially since I'm double majoring in neuroscience and biology. And I really wanted to focus on something that's related to both of it. Like it has a biology aspect and it has a neuroscience aspect. And I'm so happy that I could finally do it, thanks to Dr. Shinoy.
Good. It's great to be able to have so many choices.
Yeah.
Yeah, I think so too.
I think someone asked the question.
Okay, Dr. Cooper has a question for you. Says, so lead PB2+, I think that's what it says,
Yeah.
can block many channels, such as calcium channels. So maybe actions such as blocking all I can't even talk this morning, synaptic transmission. It would be interested to test on many types of synapses, and glutamate, like you mentioned.
Yeah. So what lead does, as you said, since it can actually pass the blood-brain barrier, it can impact a lot of stuff, as you said, Dr. Cooper. What I would be interested in to know after this with that, would there be any positive impacts of that? Just like carbon monoxide, that it had some positive impacts when rats were exposed to low levels of it. Would there be any positive impacts associated with low levels of lead? That's something interesting to know.
And then Dr. Shenoy asked if you could do a lab study on something related to this research, what would it be?
Yeah, as I said, I would have rats and some rodents that are similar. They have the similar central nervous system to humans, and I would have them exposed to low levels of lead and carbon monoxide and see myself, like, and probably PB, to see what happens in their brain. I would be really interested in that. And I see a question by Dr. Nicole Marguerite, and yes, I would love to do research on this topic. I would be really interested in that. I wish there was a lab-based opportunity for me at UK, but we'll see.
It opens doors for other opportunities for sure. Right. No more questions. Thank you very much. Good job.
Thank you.
And our final presenter today for this session is Nicole Marguerite. You may begin to share your screen at any time.
Awesome. Thank you. All right. All right. Hello, everyone. My name is Nicole Marguerite, and today I'm going to be presenting on the effect of temperature on heart rate for SERICATA and Drosophilia melanogaster with altered expression of the TRPA receptor. And so before I went into the specifics of this project, I wanted to give it a little bit of background because it seems oddly specific to be studying one specific receptor in the heart. But where we began was studying just the effects of temperature on behavior in the blowfly versus the Drosophila fly, where we see that this blowfly is shown on the left-hand side actually exhibits this penguin huddling behavior to the point where it'll push that lid off of the disc when you put them in a cool environment. In this cold environment, Drosophilia do not exhibit that same behavior. They're just wandering around. So we started to hypothesize that this could be a result of our transient receptor potential channels, which are just going to be different channels that are responding to thermal, mechanical, or chemical stimuli. And so when we're looking at temperature, we're specifically going to be focusing on the TRPA1 receptor, which is going to be responding to thermal sensation. For this, it's really interesting that these TRPA1 receptors are expressed throughout the entire body in neurons that aren't dedicated specifically for sensory modalities. So one of those areas is going to be the heart, as shown in this upper right-hand corner. It expresses many of those many of these receptors, and it specifically does express that TRPA1 receptor. So we wanted to continue to study why this might be the case. So when we're looking at those thermal receptors in the heart, we know that temperature increases metabolism in general, but we don't necessarily know how at the actual cellular level. And so we think that these TRPA receptors might play a role in that. And because we do know that TRPA receptors are, their expression is changed in cardiac pathology, it does lead to a plausible mechanism to impact that rate of heart beating. And so to actually study this, here's what the methods looked like. We had our objective camera coming down from above, where we had the dissected larva or the whole larva on our sample. And they either were overexpressing these TRPA receptors, served as a control, or they were knocked out of those receptors using an RNAi line. So we were either studying the Drosophila or the blowfly at third instar larva. So here's what it actually looked like. You can play the video. This is what all of the data looked like with these two heart tubes beating right here. We went in and manually counted these heart rates for all of our videos for those first 30 seconds. And when we get to our first results, what I wanted to show you guys first was the Drosophila for our whole larva. I think the data is relatively clean overall, and you can see a fairly nice pattern where all of our parental lines, as well as that knockout line, showed that increase in heart rate, and then it came back down when we returned those larvae back to our 20-degree environment. The big difference comes when we have that TRPA1 overexpression, where the larva is not able to maintain that heart rate anymore. Similar patterns are seen here with the dissected larva. I know this looks like a lot on this page, but we can divide it to the left-hand side is testing at a 30-degree environment, and the right-hand side is just testing at a 37-degree environment. And overall, the patterns that we should be focusing on are how this TRPA1 line we see at 30 degrees, it's not able to maintain that heartbeat. Same thing for that 37 degrees. Whereas all of these other lines are able to maintain a heartbeat at 30 degrees, and they're able to maintain a heartbeat at 37 degrees for about the first minute and a half, but then we do see a knockdown at the end. What's interesting here is at this 37 degrees, if you are to add serotonin on top of these dissected larvae, we know that serotonin is going to cause a boost or an increase in that heart rate. And so what we saw is that with either our background parent lines or the RNA line, we were able to have that heart rate increased again with application of serotonin, but we were not able to see an increase in heart rate with the TRPA1 line when it was just at that 37 degrees Celsius. What that implies is that we are looking at implications of calcium homeostasis within these cells that are being affected. Finally, I wanted to show you guys what the blowfly larvae did, because this is where our study started, looking at the differences in their behavior, which could be a result of differences in their hearts beating. And so we see that the blowfly larva is able to actually maintain this heartbeat at 37 degrees Celsius, which becomes interesting considering these larvae are going to be raised and grown at a higher temperature than Drosophila. This slide really quick is just showing that we were able to confirm that TRPA was actually overexpressed as we see this depolarization with these TRPA receptors here in those body wall muscles. So what this actually implies or means, we're trying to figure out and understand if the TRPA1 receptors have an influence on heartbeat, and if they do, how it's actually influencing heartbeat. So what we ended up finding is that the heart cannot beat normally or synchronize with this TRPA1 overexpression when we have those increased temperatures. 24 hours after the stimulation, all of the TRPA1 overexpressed larvae died, and then our RNAi and our background lines were able to survive. And then this brings it back to that point with our application of serotonin. There is a very good chance that because TRPA1 is going to also be a positive ion channel, that means that as these TRPA1 channels are opening, it's enabling more calcium to come through those channels, so the cell could be saturated with calcium, which should be why applying more serotonin, the heart was not able to beat more, considering the heart beats on a calcium cycle. So looking at some future directions, we would just continue to look to see if there's other TRP expressed in the heart for stimulation. We know that the genome in general contains about 16 different types of TRP channels. So it'd be interesting to go in and see what channels are actually expressed in the heart, as well as in other places. And then finally, looking at other thermoreceptors or looking at other species of the Drosophila. And just some references. And yeah, if anyone has any questions, I would be happy to answer those. Thank you.
Thank you, Nicole. That was really good. I like that. Let's see if we have any questions for you. I believe we do. Dr. Cooper, He says, fantastic presentation, first of all. And so do you think maybe blowflies can handle heat stress better out in nature than fruit flies?
There'd be a very good chance that they'd be able to handle that heat better. What I find more interesting is that these blowflies are known to go to dead carcasses. So when you see these dead carcasses on the side of the road, you see all the blowfly attracted to them. So it could have more of an implication of they're able to find sources of heat better than these blowfly are able to, which also could be a result of being able to maintain their heart rate at these higher temperatures as well, which could connect back to the fact that these blowfly are going to be grown at a higher temperature to begin with.
Okay, while we wait just to see if somebody else pops a question in the Q&A, tell me, Nicole, how you got involved in research and what your favorite part has been, minus this year.
I started my freshman year through the Honors College, just doing a Bio 199 with Dr. Prendergast, and I immediately fell in love with the idea of research. And right after that, I started working with Dr. Cooper. I think this is my 4th semester now working with him, and it's just been an amazing experience. I think I've been co-author on 2 published papers. I just published my first paper as primary author in January, which was this paper actually, which is very exciting. And I'm super excited to be doing research in chemical engineering next semester with the Dr. Zach Hilt. So lots of exciting opportunities lined up. And it's really just a lot of fun, especially at UK, to work with different people and continue to build your network.
Good deal. Thank you. That's great. It's good to know we have so many options here. So Dr. McLetchie asked, do you think that there can be a variation in TRPA1 among individuals within species?
Within individuals among species?
I think that's what he's asking, yes.
So I believe yes, because some of these newer papers that are coming out are showing that TRPA1 is expressed differently, just even within mammalian hearts, because of different cardiac pathologies. And so I think that these TRPA1 receptors are able to be over or underexpressed within our body, depending on what specific pathologies are going on. So I think that's a very likely, very possible.
Okay, Dr. Cooper says great answer, and Dr. McLetchie says thank you. It's a good job. I think that brings the session to an end. Congratulations, everyone. You did a great job.